Thank you very much for selecting my abstract for a talk today. My name is Strelez. I am at the Ellison Institute, and I am the organ-on-chip team manager for the Institute. We've been focusing for many years now, as I was a graduate student, developing a colorectal cancer on a chip model system. I'm going to be talking about two things today: our new patient-derived organoid model system that we spent the last few years working on, and then highlighting an example of how we can use these patient-derived organoids as a case study for a mechanistic better understanding of cancer progression. We are very interested in the tumor microenvironment here in the Mumenthaler lab at the Ellison Institute. We're really interested in the different cellular milieu of endothelial cells, immune cells, cancer-associated fibroblasts. We're also very interested in the physical forces that are very present in the colon cancer specifically when you're thinking about the peristalsis, the mechanical motions that actually move food through your digestive tract. And we're using the Emulet organ-on-chip system to better study a lot of aspects of the tumor microenvironmental complexities. The first study that I'm going to be talking about today was really, again, building up our patient-derived organoid on a chip platform. And so what we were able to do, we were able to get access to colon cancer patient tumor tissue, where we can derive colon cancer organoids from that patient tissue. We can put them on chip, expose them to endothelial crosstalk with the bottom channel with our HiMac, our intestinal endothelial cells. We can then have fluid flowing through for a course of 6 days, also have this peristaltic motions happening for 6 days, and then we can harvest the cells from that chip, and at all points of this time, we can perform RNA sequencing to understand what's happening with the tumor, the organoids pre and post organ-on-a-chip. In addition, this is a microfluidic system, so we will be having effluent collecting across time points of this experiment to understand some of the metabolomics that's happening with these patient-derived colon cancer models. We were able to, from our extensive biobank of colon cancer organoids, we were actually able to make 5 models with these 5 different patients across several different stages of colon cancer with various different tumor locations, as well as, importantly, for the later half of this talk, the KRAS status of the tumor. So we have some KRAS wild type, we also have some KRAS mutant, which will be important later in the talk. And you can see here the heterogeneity morphology of these organoids in the dish, but also on chip here. And even though we do see some of that heterogeneity across patients, we are able to form a good barrier by day 0 or day 1, depending if you're this UK organoid up here. And you can see the presence of good VE cadherin and ZO1 forming that nice barrier across all of our organoids. So one of the first things that we wanted to ask ourselves was, is this model good? Is this a good representation? Is this any better than organoids? That's a very complicated question, as many of you in this room know. That's not a simple answer, but we did do the RNA-seq across the 3 different model systems, the tumor, the organoids, and the organoid on chip, and compared the gene expression to see differentially expressed genes across the different platforms. And so we'd have the organoid versus the tumor, the organoid versus the chip, and the chip versus the tumor. These 2 comparisons here in blue and green are the important ones. So as you can see here, the chip stretch versus the tumor has a significantly lower number of differentially expressed genes than the organoid versus the tumor, starting to highlight that we are getting closer to the tumor with this chip system. In addition, we also wanted to ask ourselves, what are we missing with these model systems? And so what we did is we took the genes that were up or down in the organoids versus the tumor and the chip versus the tumor and did an over-enrichment analysis of those genes through the David platform and looked at the different pathways that were enriched in these scenarios. And as you can see, we have the genes that were higher in the organoids and organ-on-chip, and then the genes that enriched in certain pathways that were higher in the tumor. And as you can see here, to our surprise, it wasn't anything drastic that we were missing. We see a lot of ECM organization, which makes a lot of sense, as we are not able to, in this model system, recapitulate the exact ECM of these different patients. There is tumor-to-tumor variability there. We also see a lot of immune responses. In this version of the chip, we do not have immune cells yet. And we do see some different protein translation and transcription, which, again, model system differences there. And then if we zoom back out a little bit, we can start to see the differences in the patient level heterogeneity with our two different assays. So the RNA-seq from the cells on chip, you can see the different patients are clustering in this PCA a little separately, a little apart from each other. And we see the same thing with the metabolomics-based, the mass spec-based metabolomics that we performed off of the effluent from the chip. You can see that the patients do have a little bit of a spread, suggesting that we might be able to use this model system to get at some of this patient-level biology and better understand it. And before I go into the cool biology that I always like to call it, I just want to have a brief word that we are working on other model systems. Back in my day as a grad student, I was using this model with colon cancer cell lines. We've now expanded into the patient-derived organoids. We do have the patient-matched cancer-associated fibroblasts that we are also layering on to better understand the stromal tumor crosstalk. And in addition, we just started with these immune cells, adding in the PBMC components to this model system to really start to understand all the different crosstalk with the different cellular milieu. And so I want to end with a little bit of a teaser. It's a quick story with a lot of different work, so I'm happy to answer questions, but just to really highlight an example of the impact of physical forces on the colon cancer biology. This is a very unique aspect of the particular organ-on-chip model that we're working with, is that we can start to mimic some of that paracelsus. It's very understudied within the colon cancer space, but we do know that we're starting to understand that it is becoming important. One of the first things that we did way back in the day when we first started working with these organ-on-chips is develop an imaging-based invasion assay to really start to understand the first steps of cancer progression, the invasion, the tumor cells invading into the endothelial component. And so what you're looking at here is a nice worm's eye view down the channel at a specific field of view. I have my green tumor cells here in the top channel, and then I have a nice tube of RFP HUVEX in the bottom. For this particular experiment was HUVEX that we used. And then on day 0, all the green tumor cells are in the top channel, but as you can see by day 6, we have a nice cluster in the bottom of the endothelial, and you can see that they start to invade with nice single cell into the bottom channel. We can actually quantitate that based off of measuring the fluorescence of these GFP tumor cells. And one of the first questions that we asked ourselves was, what happens when we do have this peristaltic motions, this stretching capabilities of this chip to the invasion? And so we measured invasion with and without this stretching capability across a few different cell lines. And as you can see, I think the best example is the HGT woman 6s here in this black box, white black box. You can see that in the presence of peristalsis, you have a significant increase in the amount of invasion that's present. The same effect was also shown with the SW480s, and then we have the HT29s and the KCO2s down here. The KCO2s showing a non-significant increase in invasion. So we have quite a spread across our cell lines, and so we wanted to look into that a little bit further, but for the most part, for some of these cell lines, the peristalsis is really increasing our invasion. We then wanted to look at what happened in our patient-derived organoids. Once we had this model all up and running, we had the RNA-seq analysis of the stretched versus not stretched on chip. And so we compared the RNA-seq, the gene expression of the stretched versus no stretch. And you can see one of our top hits was this epithelial to mesenchymal transition is enriched in this stretch versus not stretch condition. We further broke that down by the KRAS status of our tumors, of our organoids. And you can see that in the KRAS mutant organoids, you have a highly, highly significant enrichment of this EMT pathway, whereas in the KRAS wild-type organoids, you do not have the same enrichment in the stretch versus not stretch. Again, suggesting that while we see this nice phenotypic increased invasion in the peristalsis, in the increased invasion due to peristalsis, we're also seeing this nice indication with the EMT from the gene expression. We then, of course, have to look on chip to see what the cells are doing with their invasive capabilities. And so what we did here, we were able to GFP label 2 different of these patient-derived organoids. And you can see here in this particular organoid. These are my US organoids, which happen to be KRAS mutant. You can see that there's a nice increase in invasion in the stretch conditions, whereas in the UP organoid, this is my KRAS wild type organoid, we do not see that increase in invasion due to the stretching, suggesting that this is potentially a KRAS mutant crosstalk with this mechanical forces. Furthermore, we're able to collect some of the circulating tumor cells from this chip. So these are the cells that have invaded from the top channel and then end up in the endothelial effluent. We can collect them. We can do various assays with them. They remain viable. And we did some RNA-seq of the stretch versus not stretch circulating tumor cells to better understand and validate what was going on. And so you can see a lot of cell adhesion, integrin, cell migration. Again, awesome, we're validating this phenotype that we see on chip of this increased invasion. But we also saw this regulation of GABAergic synaptic transmission, which was pretty new to us. We didn't really think anything of neurotransmitters within this space. And since we do have access to all of this effluent that we're collecting over the course of the experiment, we decided to look into the mass spec-based metabolomics to try to measure GABA to see what was going on, to see if this gene expression changes of the GABAergic signaling also resulted in more of GABA. And so what you can see across all 5 of these patient-derived organoids, we're actually measuring GABA levels in the top channel effluent over time. So I'm measuring my day 6 and my, day 0 and day 6. You can see that there is GABA present in the effluent of all of these chips, but in some of the chips you can see a nice increase on day 6, suggesting that we're getting more GABA in the effluent over time. This is also, if everyone's catching it, this is the US organoid, which was also the KRAS mutant organoid that showed nice increase in invasion due to this mechanical forces. We looked into what on earth the connection with GABA and cancer was. And it's actually an understudied but known phenomenon that GABA can be connected with cancer proliferation and metastasis, with exogenous GABA being present, having a lot of signaling consequences resulting in increased proliferation and metastasis. In addition, a lot of interesting immune connections, which hopefully we'll get to in future studies. But before we get too excited about this, 1 or 2 different patient-derived organoids that did show this GABAergic signaling, we wanted to look into the clinical data as well. And so what we did is we went to the TCGA and looked at GAD1, which is the enzyme that makes GABA from glutamate, to see what's happening in these colon cancer patients with this GAD1 expression. And so here I'm showing colon cancer patients, primary tumor, so no metastatic patients at this moment. And we're looking at GAD1 expression. Because we had this KRAS hypothesis, we did look at mutant versus wild type patients, tumors that had KRAS mutant versus KRAS wild type. And you can see a highly significant increase in expression of GAD1 in the KRAS mutant tumors. Furthermore, we also looked at survivorship, and so we looked at, in these KRAS mutant patients, those patients that had high levels of GAD1 are in red here, and had worse outcomes than those that had low GAD1 expression, giving us a little bit more power to this hypothesis that there's something going on here with the GABAergic signaling. And furthermore, there's a lot more data. I'm happy to answer more questions, but to make this story fit here, what we ended up doing is getting to this GABA catabolism in these tumor cells. And so ABAT is the enzyme that catabolizes GABA and kicks it into the GABA shunt into the TCA for metabolism. And we can do some pharmacological inhibition with these patient-derived organoids. And so you can see here, when I have this ABAT inhibitor, this bigabatrin here, in the stretch conditions, we see a nice decrease in the invasion, the Paracelsus-mediated invasion. So there's nice crosstalk here happening with the KRAS status, invasion, and progression, and this GABA signaling. Again, we have a lot more work to do. We're hoping to expand this out. But this is a case study of how you can use these chips and the physical forces that come along with this particular organ on a chip to start answering some new mechanistic studies. And so to conclude here, we have been working really hard to get a patient-derived organoid model system to study the tumor microenvironment, the role of physical forces, particularly in KRAS mutant tumor cells, to study cancer progression, and the concept that this organ on a chip with the patient-derived organoids can really recapitulate important aspects of the patient tumor microenvironment, including neurotransmitter signaling, in this example, GABA. And so I would like to quickly thank, like I said earlier, I'm in the Shannon Mumenthaler lab of the Elson Institute. This was funded by NCI R01, awarded to Shannon Mumenthaler. This is a highly collaborative project across the Institute and across the University of Southern California. A lot of mass spec involvement, so thank you to Jonathan, Katz, and Jack. And then my team, the Organ on a Chip team, is a phenomenal, small team right now, but really hardworking team, and all of their data and all of their efforts was a part of this presentation. I couldn't do it on my own. And so I'm happy to take any questions. Thank you.
Thank you very much, Carly. Are they? Oh, I already see a questioner run. Because we are short on time, please be precise.
So how physiological are your stretch forces if you compare it to the real situation? And how did you calculate that?
What was the last part?
How did you calculate that?
That's a great question for Amulet. So Amulet has done a lot of those studies, I believe, and they have done all the studies in guinea pigs. I believe it was guinea pigs to study the physical forces of the peristalsis. We haven't really adjusted much of the peristalsis, like less or more of the deformations, but it is coming from some animal work to inform the forces here.
I have a question, but I think it's better from the audience, so it's fine.
I just have a question about how you plate your cells, because these are organoids that grow in 3D, but in your chip they're now in 2D. And when you look at your gene profiles as well, you see especially things like adhesion proteins and ECM proteins are now altered. So how closely do you actually think this is to in vivo tumor?
Great question. Definitely one of the technical challenges of these organoids and getting them on chip. We do fragment them down quite a bit to get them on chip. So absolutely not are we able to really recreate the full 3D construction of the tumor. However, the benefits of all the crosstalk with the other cells seems to be important for us. So great point and important considerations.
Okay, so thanks a lot, Carly. I think we have to move on. So thanks again.